Hello everyone, my name is David, and today I'm going to introduce you my topic, Sheina orri, Myodocopa cypridinidate and ostracod parasitic on the gills of the epaulette shark, Hemicillium ocellatum elasmobranchid hemicydate. And this paper is from International Journal for Parasitology. Firstly, we need to know what is ostracod. Ostracod is in a subphylum of Crustacea, so it is a crustacean. And there are 70,000 species in ostracods, but only 13,000 of which are extant. Extant means something very old but is still existing. And ostracods are very small, typically around 1 millimeter in size. Their body are flattened from side to side, protected by a chitinous or calcareous shield. And then I want to introduce you cypridinidate. Cypridinidate is a family from ostracod. And the more important thing is that we need to know their feeding strategies. In general, their feeding strategies can be divided into 3 different types: collectors, predators, and scavengers. Collectors likes to eat small particles of detritus from sediment. Next, predators. Predators are good at swimming and they are greedy carnivores. They like to eat large active prey such as fish larvae and copepods. Lastly is scavengers. Scavengers feeding on large particles of debris and the remains of the fishes. Scavengers do not attack the healthy fishes, but there are some exceptions. For example, they will attack the fishes which confined in the cage at night. So, what is the big deal? 3 authors reported case of so-called parasitism in cyprinids, and also none of previous study of possible parasitism in cypridinidate have investigated the attachment of the ostracods for its effect on the host tissue. So we re-examine the occurrence and distribution and compare the feeding appendages of ostracods employing other feeding strategies. Appendages in invertebrate biology we call appendages, and in vertebrate biology we call limbs. To complete this experiment, firstly we need animals. 28 adult epaulette sharks and other ostracods used to compare the feeding appendages collected by dead fish trap and midwater trail. Secondly, statistically posture. The distribution of ostracods on the gills of epaulette sharks was examined statistically, and we used Kruskal-Wallis one-way analysis of variance on ranks. Last one is specimen preparation. Firstly, we remove the gills, placing in 4% of formaldehyde in seawaters. And then examination of each hemibranch identified as Sheina orri, the species we want to observe. Next, a few ostracods were collected for light or scanning electron microscope. Lastly, additional ostracods were used to investigate morphological differences. And in the results, we can see from the 3 aspects. The first one is histology. As you can see in this picture, which shows the epaulette shark scale filament. And letter P means groovies on shark scale filament. Groovies is like holes on the shark's scale filament. There are many holes. And blood cells, letter B means blood cells and tissue debris. Blood cells and tissue debris of shark origin were often present between the two carapace valves of ostracods. Carapace valve means the left valve plus the right valve. And the ostracods will grab the gill filament, cause these groovies, but the mechanical damage caused by the attachment and feeding activities of Sheina orri is minor. And next, we see the picture on this slide. Picture A, picture C, and this picture D. All shows the folds in the interbranchial septum. The fold was a pre-existing structure. My answer is we couldn't know. Because, as I just mentioned, the mechanical damage caused by the attachment and feeding activities of Sheina orri is minor, and there is no response or little response to it. So I hope I will convince you that we couldn't know these folds are a pre-existing structure used as an anchoring site or just caused by the ostracod's actions. And secondly, we compare with other ostracods feeding appendages. As you can see in this picture, shows Sheina orri's maxillular and mandibular cloud. Sheina orri's maxillular cloud is highly curved and is large. And in Sheina orri's mandibular cloud, you can see the pad morphology. And then we compare with scavenging ostracods maxillular and mandibular cloud. Scavenging ostracods maxillular cloud has pectinate teeth on the claws, and its mandibular cloud didn't have the pad morphology. Also, predatory ostracods mandibular cloud didn't have the pad morphology too. And it's just slightly curved. So it is unclear to how pectinate teeth absence would enhance the attachment to the gills, and which might be an adaptation to parasitism is the pad morphology at the tip of the middle seta. Therefore, the pad morphology will form a pincer and allow the ostracods to collect the material on the gill surface or grip the gill surface, grip the gill filament. Sorry. And that's why we observe the life cycle of Sheina orri. We just found the adult ostracods Sheina orri on epaulette sharks gills instead of the juvenile ostracods. The absence of the early instars indicate that Sheina orri might just parasitic for part of its life. In conclusion, it is likely that parasitism within the Cypridinidae has evolved as an extension of scavenging. Although some of the functional morphology in the maxillular or mandibular cloud of Sheina orri might be suited to parasite, but Sheina orri and Cypridinidae isn't the only one who suit to parasite. And here's my reference, and thank you for everyone.